Hi, Miss Nikki here. Welcome to Chapter 24. We talked about inheritance in the last chapter, looking at Punnett squares and genotype and phenotype. Now we're going to kind of look at it from the chromosomal aspect. So we'll look at some things like gene linkage. We'll look at how certain genes are carried on specific chromosomes, like X-linked. We'll talk about some non-disjunction events that can happen with chromosomes. So during meiosis 1 or 2, you can have chromosomes that don't divide properly, and that this can lead to some patterns of inheritance that we'll see, or some disease states. Here we have an example of genes on chromosome 19. They want you to see that genes are fixed, or that they're in a specific order, because some genes are so closely linked that you're always going to see them expressed together. So this gene, when expressed, will always be linked with this gene expressed. So this is where we get this term gene linkage. So during meiosis 1 and 2, these genes are so close together, there's not a very good likelihood that they're going to be separated. So you'll also see them talk about the fact that these are non-independent assortment. Remember how the chromosomes line up on the metaphase plate and you never know which chromosomes? We talked about independent assortment in the last video. They're saying that these are not independently assorted. They're always going to be carried together, or more likely that they're going to be carried than not to the offspring. We have this idea of complete gene linkage. So genes A and B are so close to each other that they're inherited together. Recombination is very rare. We can see that if genes A and B are farther apart, if there's more space between them, it's more likely that a crossing over event can occur between them, and that will allow for more recombination, or recombinant gametes. We've discussed before that we have 23 pairs of chromosomes. 22 pairs are called autosomes, and those are homologous, right? One pair is called sex chromosomes. And if you're a male, you have one X chromosome and you have one Y chromosome. And if you're female, you have two X chromosomes. And I'm being very general. We'll talk about some issues coming up in just a little bit. X and Y are non-homologous. They don't have the same genes on them, right? So they have different combinations of genes. The Y chromosome, we're going to see a gene called SRY, which is basically what causes the embryo to develop male characteristics. So if mom is XX and dad is XY, and Y makes you male, if you're a son, you had to get your Y from your father, and you had to get one of your X's from your mother. If you're a female child, you got an X from your mom, and you got the other X from your dad. So we're going to look at some X-linked genetic disorders. They're always recessive, and the females must have 2 copies to be affected, where the males only have to have 1 copy. Nice chart here. It's showing you that if you have 2 X alleles, it has to be a female. And in this case, they have both genes. The big B will be normal. They have 2 genes for normal color vision. The lowercase b will be color blindness. So here are the 2 X alleles. That's a female. She has 1 normal and she has 1 diseased allele. She is a carrier. Then you have a female that has 2 colorblind alleles, so she's colorblind. You can see with the male, since he only has 1 X, if he gets a good copy from mom, great, he has normal vision. If he gets a diseased allele or a bad copy, he's going to be colorblind. So these X-linked alleles seem to affect males more because they don't have the ability to mask the recessive colorblind allele with a dominant normal vision allele. So females kind of have redundancy. We don't see as much color blindness. Males seem to be disproportionately affected because they're only getting 1 allele from their mom. You can see that this carrier, we now then have a 50-50 chance of passing that allele on to our child, right? we could go back and do that test cross and look at the Punnett squares for the possible offspring and then look at percentages. So here is a male and he has normal vision. And here is a female and she's a carrier. So mom can donate this X or this X, and dad can donate this X or this Y. So you can do the down and over, and you can look at the possible offspring. So they have a chance of having 3 phenotypic, right? So 3 out of the 4 will have normal vision. We have 1 out of 4, or a 25% chance of having color blindness. Now, if I'm looking at genotype, that frequency is a little bit different, right? So genotypically, I have 1 female that has normal vision. I have 1 female that's colorblind. I have 1 male that has normal vision and 1 male that's colorblind. If you were looking at a pedigree for an X-linked disorder, you'll see that males are more affected or disproportionately affected compared to females. It's much less likely probability-wise that a female would receive 2 diseased alleles. She'd have to get 1 from her mom and she'd have to get 1 from her dad. X-linked dominant disorders are very rare. This would be only 1 copy needed to cause disease. So only 1 copy. If you're a male and you have this dominant disorder, 100% of your females or offspring are going to have this disorder. Because remember, the Y is going to go to your male offspring, the X is going to go to your female, and you only need 1 copy. So if you see an X-linked dominant disorder on a pedigree, all of the daughters are going to have the disorder from the male who also had the disorder. Another X-linked recessive disorder that's somewhat common is Duchenne's muscular dystrophy. Basically, it's due to a lack of the dystrophin protein. And that protein helps kind of your nervous system talk to your muscles. And so over time, the muscles start atrophying. Another X-linked recessive disorder is Fragile X syndrome. It's caused by an abnormal number of repeat sequences, specifically CGGs. So if you increase the number of these repeat sequences that aren't supposed to be there, you can imagine that that would interrupt protein translation. So remember, we have DNA codes for RNA, and then that codes for our protein. So this is going to disrupt some of the protein products that we can make the more repeats we have. So this is why you can see a range from mild to severe. It depends on the number of repeats. And also we'll see this increase through generations. So it seems to get worse as we pass on these repeated sequences, and maybe they repeat again. So sometimes we could have over 200 repeated CGG sequences, and then you would have more severe intellectual disabilities. Another X-linked disorder is called hemophilia, and we know a little bit about this because it passed through European royal families. Hemophilia is a blood clotting disorder. So basically, there are certain factors that help your blood clot if you have a cut, and they are missing some of these factors. So either the blood doesn't clot, so no clotting, or very slow clotting. So you can imagine if something were to happen to you and you had a gash, that you might bleed out before the clotting process was finished. And something to notice, because this is X-linked, and we said X-linked mostly affects males because they only have to have 1 X copy to have the disorder. So you can look at this pedigree and see that all of the males were affected. This is an example of a karyotype of human chromosomes. So we can take a sample of your blood and we can sort the cells out that have a nucleus. So your red blood cells don't have a nucleus, so we don't want to use those. But we can sort out the cells that have a nucleus, and then we can trigger division and arrest the cells in the middle of division so that we have our nice chromosomes lined up. And then we have computer programs that will sort out these chromosomes based on size and based on specific banding patterns that we use when we stain the chromosomes. So we're seeing here the 22 autosomes, and then we're seeing the pair of sex chromosomes. So we're supposed to have 46 chromosomes, right? Well, sometimes we can have different numbers depending on if meiosis 1 or 2 did not divide properly. If chromosomes do not divide properly during meiosis 1 and 2, we call this a non-disjunction event. As a result, some of the gametes are going to have an incorrect number of chromosomes. I'm sure we've heard of Down syndrome. Down syndrome is trisomy 23, where we have 3 copies of chromosome 23. There's also monosomy, where you only have 1 copy. Nice example here for non-disjunction event. So if the non-disjunction happens in meiosis 1, you can see your pair of homologous chromosomes don't separate evenly. So we have 4 chromosomes here, we have 0 here. This side, if you divide 0, you get 0, right? And then we have the sperm that's going to fertilize the egg, so we end up with only dads. What happened to moms? It's missing. So we have 1 less. This is monosomy. If we follow the other side, we had 4 chromosomes. During meiosis 2, they divided normally. In comes the sperm, but now I have 3 of the same chromosome instead of 2. So this was 1 extra. This is trisomy. You can see if the non-disjunction event happens in meiosis 2, it's a little bit better. So here's our pair of homologous chromosomes. They separate normally during meiosis 1. This cell continues, this daughter cell continues on its normal journey, and we end up with the correct number of chromosomes in our zygote. But if the non-disjunction event happens in meiosis 2 and it's only affecting this daughter cell, here's where you end up with this daughter cell has 0, this one has 2, then we add 1, that gives us 3, or trisomy, and we have over here -1, which gives us monosomy. But you can see that you have a 50-50 chance of having normal offspring. So a meiosis 2 non-disjunction event is better than a meiosis 1 non-disjunction event. If it's a meiosis 1, all gametes are affected, and all offspring, if you want to say, zygotes as well. If it's a meiosis 2 event, you've got a 50-50 chance. Normal development depends on exactly the right number of chromosomes. So we need 2 of each chromosome for a total of 46. If we deviate from that number, we're going to have problems. We're either going to have too much protein production or too little. For trisomies, the only ones that survive to childhood and adulthood are Down syndrome, trisomy 21. Trisomy 13 and 18 have been born, but they rarely survive past the first few months. And this is not to say that these are the only chromosomes where you can have non-disjunction events. You can have trisomy chromosome number 1, but the effects would be so dramatic and drastic that the child would not make it to delivery. They would not be born. So probably some pre-spontaneous abortion event would occur. Here's a nice table showing you the changes in chromosome number and then the syndrome name. We just talked about trisomy 18, 13, and 21. There are also some cases of monosomy or trisomy with the sex chromosomes. We can have changes in sex chromosome number like we just saw on the table. If your mom was X X and she donated an X to the egg, and dad was XY, and there was some sort of non-disjunction event where the sperm arrived and it had no sex chromosome. So the sperm is going to fertilize the egg. The other chromosomes were present, the other 22 chromosomes. So this zygote, this fertilized egg, will turn into or develop into an embryo and a fetus and be born, but they're missing a sex chromosome. So the child ends up having X, and you could say blank. Some people write XO to say that that's a missing sex chromosome. This syndrome is called Turner syndrome, and they are always female because remember the Y chromosome has that SRY gene that I talked about earlier, and that's what triggers the development of the embryo into a male. I wanted to mention Jacob's syndrome, where the males have an extra Y chromosome. We used to think that this extra Y made males more aggressive, but we've come to study and realize that there is no behavioral difference. The problem was they were looking at males with the XYY that were in prisons or they were in mental institutions. So this is just a good example of you have to be careful where you get your data from, and that your data has to be random or you're going to make inferences based on biased data. So there can also be changes in chromosomal structure. So it's not missing a chromosome or an extra chromosome, it's some sort of chromosomal mutation that we're either going to have a deletion of a gene, we're going to have a duplication of a gene, we can have a gene that's been moved from its original location, or we can have a gene that's inverted, it's upside down. And all of these mutations can lead to syndromes in the offspring. Here we have an example of a deletion, Williams syndrome. You can see that the tip, the distal end of this chromosome has been lost. So if a child has this deletion, they'll have a very similar phenotypic presentation. So sometimes if you're a clinical geneticist, you can look at a child and suspect if they have a certain syndrome based on a deletion, an insertion, a translocation, because the phenotypic presentation is very similar in case to case. Here we have a nice example of an inverted duplication. So most of us understand duplication. If we had D and E, and then here we had D and E repeated again, that would be a duplication. You can see that this chromosome is longer in length. So we have, from those genes, we're going to make proteins. So now we have increased or high amounts of protein for that particular action. And here we have a duplication, but then we also have inverted that duplication. And you can look over this list. This is an inversion duplication 15 syndrome. It causes poor muscle tone, mental impairment, seizures, curvature of the spine. So also keep in mind, chromosomes don't just have one function, right? There are several hundreds of genes on chromosomes, and each one is going to have a pathway that affects a different part of the body. Here we have an example of a translocation where this is chromosome 2 and this is chromosome 20, and in this translocation event they've managed to swap genes. So now we have the wrong gene on the wrong chromosome, and this is going to have some sort of biological or physical effect. I wanted to show you kind of compounding events that could happen. So let's say that during replication, when the DNA was copying itself, so replication, copy DNA. When the DNA was copying itself, this inversion took place. So this should be C and this should be E, but they've inverted, right? They're upside down. And then this cell with these chromosomes are undergoing prophase 1, and that crossing over event that takes place during meiosis. Now we have a complete reshuffling of genes on these homologous chromosomes. So we have both duplications and deletions. You can see over here we have A and B. Well, we also have A and B down here we're not supposed to. And then if you look on this other chromosome, you can see that we have duplication, G and G, and F, but we're also missing A and B. So if you have these kind of compounding events where you have some sort of chromosomal structure issue, and then you have this kind of crossing over event where we're trying to have recombination in our gametes, it can lead to total chaos with both duplications and deletions. All right, so that's it for chapter 24. I'll see you guys next chapter. Bye.